Welcome back to the deep dive. If you've ever tried to understand a large crowd by focusing on just the average, you miss all the interesting stuff.
You miss the rare interactions, the individual choices.
Exactly. And in biology, for decades, that's kind of what we've done. We look at billions of molecules in a test tube, and, well, we just get an average reading.
An ensemble measurement.
Today, we are strapping on the metaphorical single molecule microscope. We're diving into single molecule biophysics, where we stop looking at the average and start watching individual biomolecules.
Things like proteins or DNA doing their jobs in real time.
Now, this field is powered by some amazing techniques like single molecule fluorescence, SMF, and also Forster resonance energy transfer, or fourth partella.
And they're phenomenal. They let you detect those rare, fleeting events that the ensemble measurements just blur out completely.
Plus, they need next to no material, which, as you know, is a huge blessing in biological research.
Oh, absolutely.
But there has always been this massive, painful bottleneck. To compare, say, a healthy enzyme to a drug-resistant mutant, you had to run one experiment, clean everything up, set up the next one.
And then run it again. You were always stuck looking at one sample at a time.
And that limitation introduces so much noise.
An impossible amount of noise sometimes.
Yeah.
If you ran 4 experiments over, say, 2 days, the subtle differences you were trying to measure, maybe a tiny change in how a protein folds,
They could just get washed out.
completely masked by tiny fluctuations, buffer pH, laser intensity, even just slight changes in the surface you're using.
You might see a difference, but you couldn't be sure if it was your mutant protein or if, I don't know, the air conditioning just kicked on halfway through your second run.
Exactly. That lack of simultaneous throughput was the major hurdle.
Which brings us to our sources today. We're looking at research detailing a brand new method that was engineered to just eliminate that bottleneck.
It's called parallel rapid exchange, single molecule fluorescence, and FEFIT, or for short, PRE-smFRET.
So our mission today is to really unpack how this new technology lets you interrogate diverse biological systems at the same time under perfectly identical conditions.
And we're going to walk you through the astonishing engineering that makes it possible, and then explore 4 really stunning biological discoveries that came right out of it.
Okay, let's unpack this core technology first. What makes PRE-smFRET so revolutionary? It's got to be more than just, taping 4 microscopes together, right?
It's all about the integration. The real revolution is achieving these parallel measurements that are, and this is key, a priori.
A priori, meaning you know the identity and the location of the sample before you even start collecting data.
Exactly. And you're doing it under these rapid, kinetic, pre-steady state conditions.
So no mixing 4 samples in a pot and then hoping to sort them out later. You know exactly what's happening, in which pixel, and you can trigger a reaction across all 4 systems at the exact same time.
You got it. The imaging backbone they use is wide field, prism-based, total internal reflection fluorescence, PTIRF.
Which is crucial because it only lights up the molecules right at the surface, giving you that incredible signal-to-noise.
Right. And they couple this with custom-built microfluidic chips. These chips have micro-contact printed microarrays on them. Think of it like molecular scale printing.
So the microfluidics are the plumbing, and the microarrays are the reaction sites. How do they make sure that 4 different molecules, say a wild type and 3 mutants, actually stay in their designated spots?
That is the genius of it. They print 4 different orthogonal DNA anchor strands in a precise 2x2 grid, right in the camera's field of view.
Orthogonal meaning they don't interact with each other. They're chemically distinct.
Precisely. So you tag your 4 different biomolecules with 4 complementary DNA barcodes. The barcode on your molecule only sticks to its specific anchor spot.
Ah, so you can flow all 4 samples in, and molecule A only ever binds to spot A, B to B, and so on. They're physically separate, but in the exact same environment. That explains the spatial part. But what about the rapid exchange? That sounds like a serious engineering challenge on that scale.
It is a phenomenal feat. They have a computer-controlled liquid handling system coupled right to the chip. And this lets them do reproducible solution exchange like delivering a drug to start a reaction in as little as 60 milliseconds.
60 milliseconds. I mean, to put that in perspective, how slow is a typical manual exchange?
Seconds. If you're really fast, maybe a few seconds. A basic automated system might take a full second.
And so many biological events are over in a flash.
They happen on the sub-second timescale. Protein folding, ligand binding. If your solution exchange takes 500 milliseconds, you've missed the entire start of the reaction.
So the 60-millisecond exchange is like having a stop-flow instrument, but for single molecules.
It's the PTRF equivalent, yes.
Hmm.
But you're watching individual molecules, and you're doing it for 4 different systems in parallel.
Yeah.
It just opens up a whole new world of fast kinetics.
And we have to touch on this immobilization system, ARES, epitaxial removal, aided by strand exchange. You said it lets you reset the surface.
Right. Think of it like a reusable molecular Velcro.
Mm.
The molecules are anchored with DNA. When you're done, you don't throw the chip away, you flow in a special removal strand.
And it just peels them off.
It displaces the anchor strands holding the biomolecules. It's called toe-hold-mediated strand displacement. It lets you strip the surface completely clean and reset the whole device for a new experiment.
So it's not just faster and more precise, it's also reusable and efficient.
Incredibly so.
Okay, so they solved the space problem, the speed problem, and the cleanup problem. Let's get to the results. How well does this parallel approach actually reduce that experimental noise we talked about?
This is maybe the most compelling validation in the whole paper. We know single molecule data is so sensitive. Laser fluctuations, buffer aging, oxygen scavengers.
All sources of noise when you do things one after another, so they needed a head-to-head comparison.
Exactly. They used the platform to do a definitive bake-off of 8 different fluorophores, 4 donors, 4 acceptors, all at the same time in one field of view.
The ultimate control experiment. 8 things measured under the exact same conditions.
And the noise reduction was dramatic. The study found that the standard deviation of the total photon yield was at least 4-fold lower when measured in parallel compared to doing 4 separate experiments sequentially.
A 4-fold reduction in variability, that's transformative.
It means you can confidently say that a 10% change in efficiency is because of your mutation, not because the laser flickered.
And they found something huge about the dyes themselves, didn't they?
Oh, absolutely. Their parallel analysis showed that these self-healing LD555 and LD655 fluorophores just globally outperformed the more popular Sina and ATO dyes.
Especially under certain conditions, right?
Yes, specifically in deoxygenated environments, which researchers use all the time to make the dyes last longer. In those conditions, the ATTO fluorophores just dimmed substantially.
That's a substantial.
ATTO 532 brightness dropped 4-fold. ATTO 647N dropped 2-fold. And its total photon yield went down by nearly 5 times.
Wow. So they basically exposed this hidden weakness in a widely used tool just by doing a perfectly controlled comparison.
Right. It means if you were using ATTO dyes in that environment, you might have been missing a huge chunk of your signal, potentially misinterpreting your data.
A huge practical validation. So if we connect this to, say, the bigger picture of biological signaling, this seems perfect for figuring out the order of events in a complex process.
Exactly. It's ideal for resolving the order of events in protein activation, where timing is everything.
Which brings us to the first big biological system they tackled, fluorescin-1 activation.
Fluorescin-1 is a crucial protein. It's what happens after a GPCR-AG protein-coupled receptor gets stimulated. It's the protein that turns off that main signal and then kicks on alternative pathways.
And its activation involves a few moving parts.
Several. The C-terminal tail releases, Sibyl loop shifts, and the whole C domain rotates about 20 degrees relative to the N domain.
So 3 big conformational changes. And the question was, do they all happen at once, like a switch flipping, or is it an ordered sequence?
And traditional methods just couldn't tell those steps apart. But PRE-smFRET allowed for what they called multi-perspective imaging.
So they built 4 different sensors for fluorescin-1, each one reporting on a different change.
And they imaged all 4 simultaneously in the same field of view. It's like having 4 camera angles on the same molecular event.
So what did the kinetics show? What's the order of events?
Well, they found a statistically significant difference in the apparent affinity. When the activating peptide was introduced, the finger loop sensor showed a much higher affinity. It bound tighter compared to the C-tail release sensors.
Okay, so the finger loop change happens earliest, stabilizing that initial binding.
That's the interpretation.
Yeah.
But the critical kinetic finding was this. The pre-steady-state measurements showed that the C-domain rotation is likely the ultimate rate-limiting step of the whole process.
The big 20-degree rotation is the final bottleneck.
Exactly. It proves it's an ordered sequence. The protein doesn't just snaps active. It moves through these defined kinetic steps.
And the takeaway is massive. If you're designing a drug, you now know the kinetic bottleneck to target. You could aim to inhibit that final rotation, which could be much more effective.
It allows for much more precise targeting, absolutely.
Okay, speaking of complex targets, let's move from human signaling to the bacterial engine room, the ribosome.
The bacterial ribosome is a multi-megadalton machine. And the researchers focused on tRNA selection during mRNA decoding. This is where the ribosome checks if it's reading the genetic code correctly.
And they use the parallel setup to look at 4 different versions of this at
Right?
once.
2 with perfect match, or cognate codons, and 2 with near cognate codons, which have mismatches that the ribosome should reject.
And then they added the antibiotic streptomycin.
Exactly. Streptomycin is famous for making the ribosome sloppy, for causing miscoding. But the data revealed a paradox. 2 really unexpected effects.
Okay, what was the first one?
The first surprise was that streptomycin significantly reduced the apparent rate of the initial binding step, the tRNA complex arriving at the ribosome by about 2-fold. It slowed things down at the start.
It slows down entry. That seems like a pretty standard inhibitory effect. But you said there was a contradiction.
There was, because also, unexpectedly, streptomycin significantly increased the decoding rates, the steps after initial binding, and increased the overall efficiency for both the correct and the incorrect tRNAs.
Wait, I'm trying to picture that. It slows the tRNA from getting in, but once it's in, it actually helps it work better and faster.
That's a perfect way to visualize it. The analysis suggests that streptomycin promotes a tighter closure of the ribosome's small subunit, or SSU.
And that's a double-edged sword.
Precisely. The tighter closure makes it physically harder for the tRNA to get in, which slows down the initial binding. But once the tRNA squeezes in, that tight closure stabilizes the interaction and pushes the reaction forward much faster.
That is a phenomenal insight. It's not just making the ribosome sloppy, it's changing the physical dynamics of the whole process.
Something only these kinds of ultra-fast, parallel, single-molecule measurements could ever resolve.
Let's look at another layer of complexity. Natural biological variation. What did this method reveal about the fact that not all ribosomes in, say, an E. coli population are identical?
This gets right to the heart of how bacteria adapt and develop drug resistance. E. coli actually has 7 slightly different ribosomal RNA operons. And under stress, like when food is scarce, ribosomes made from one specific operon, turinators, become much more abundant.
So the bacteria can actually switch its manufacturing to produce a slightly different version of its engine.
Exactly. So the researchers compared ribosomes made from the common hydradase sequence to those made from the stress-induced Trexel variants.
And they did it side by side with PRE-smFRET.
Under perfectly identical conditions. And they found that these naturally occurring ribosome subtypes showed differential sensitivities to drugs within the same exact chemical class, the tetracyclines.
What was the specific difference?
The ribosomes with the stress-induced TARD variants were relatively resistant to the drug tetracycline, but conversely were relatively sensitive to oxytetracycline.
So resistance and sensitivity can flip just based on these tiny, naturally encoded sequence variations?
Yes. This subtle functional distinction was only provable because they could do a direct, simultaneous comparison. It shows that this ribosomal heterogeneity isn't just a curiosity, it's a kinetic factor that directly influences which drugs will work.
That's incredible. It fundamentally changes how you might think about treating a bacterial infection under different conditions.
Absolutely. So to summarize the impact, PRE-smFRET gives you high spatial and temporal resolution, it crushes experimental variance, that 4-fold drop in noise, and it lets you do these critical kinetic measurements with just picomole quantities of material.
A tiny fraction of what you'd need for traditional bulk studies.
An order of magnitude less.
So, what does this all mean for you, the listener? This technology means that incredibly complex systems that were either too challenging or required too much precious material-like signaling complexes from rare mammalian tissues can now be studied with unprecedented resolution.
We can finally peel back that complexity and reveal the subtle kinetic differences that drive signaling, that cause disease, and that determine whether a drug will actually work.
And this seems like just the beginning. The paper details 4 samples at once, or maybe 8 if you get fancy with colors.
But they show that just by changing the printing method and using a bigger camera, imaging 9 independent samples in a single field of view is already achievable.
So the transition from a specialized low-throughput technique to a true high-throughput screening platform is already happening.
The ability to scale up while simultaneously reducing noise is going to accelerate discovery immensely.
Which points to a powerful future. You can imagine a generalizable screening platform built on arrays of hundreds of different molecules, protein variants, drug candidates. You could screen libraries faster and with higher fidelity than ever before.
The possibility of discovering drug mechanisms at the single molecule kinetic level, but scaled up for practical use, it's now within reach. The speed and the sensitivity are just transforming the landscape.
We'll leave you with that thought. The era of high-throughput single-molecule screening has arrived. That's it for this deep dive.